Hello everyone, this is a continuation of the previous video that you have seen. Okay, in my previous video you have learned about the work of Mendel, that is how he did a cross on the monohybrid cross, taking the character as same height, in which he crossed the tall to the dwarf, and then the outcome of the F1 generation were different in the F2 generation. Okay, based on that, Mendel he proposed, based on his work, that is a monohybrid cross, he proposed two laws. Actually, there are three laws, but now we will discuss only the two laws. Okay, so let us see that. And the law is known as principle or law of inheritance. The first law is law of dominance. Okay, the law of dominance. In the law of dominance, according to Mendel, the character is controlled by a discrete unit known as factor. Okay, characters are controlled unit known as factor. Factor is nothing but it is a gene. Okay, characters are controlled by a discrete unit known as factor. And the second point is factor always appear in pair. For example, the gene. Factor means the gene. For example, when you took the character of same height, that is capital T, or small t, which means this is a gene. Okay, and they will always appear in a pair. And then the third point is in a dissimilar pair. In a dissimilar pair like the heterozygous. Like the heterozygous, with example capital T, small t. Okay. One dominates over the other. That is, though the small t is present, but it won't express itself. Why? Because capital T is dominating over the other. Okay, this is the law of dominance. This is what the law of dominance says, that characters are controlled by a discrete unit known as factor. Factor always appear in pair. Remember that factor is nothing but a gene. And in a dissimilar pair like the heterozygous, example is capital T, small t, one dominates over the other. Law of segregation. Okay. The law of segregation states that the factor or the alleles, they never blend or they never mix up. Okay. Alleles never mix. Alleles never blend or mix. Why? Because, for example, like in the monohybrid cross, we have seen that in the F1 generation, all the characters appear as capital T and small t. But in the F2 generation, we found differences in the genotypic ratio. That is, we saw some dwarf characters. That is the small t at the end, which means that this allele, they do not blend. Okay, the alleles, they do not blend or mix up with the other, or else the outcome would have been mid-height. The stem of the plant would have been mid-height or of any other kind. Okay, but there is no mixing of this allele. That is why the law of evolution says that alleles never blend or mix up, but it gets inherited. It gets inherited from the parents to the offspring as it is. I hope you got it. Let us see what is a dihybrid cross. Okay, the third law that I've told you, that is law of independent assortment. To learn that, I think dihybrid cross is important. Okay, so let us discuss dihybrid cross. I've already told you what is dihybrid cross, isn't it? So dihybrid cross is nothing but it is a cross made of two characters. Okay. Or you can say it is a cross made of two genes, taking two genes. Okay. In this case, Mendel in the monohybrid cross, Mendel did a cross of the single gene. That is, he took the character that is stem height. But here we can choose any other characters. Why? Because as we have seen that there are 7 characters, so we can choose any 2 characters. So to do this, let us take the character of seed, shape, and seed color. Okay. Even in this case, there will be one dominant and the other recessive. Okay, in which we will use alphabets. Okay, for the seed, dominant, recessive. Okay, for seed shape, we can use round. For dominant, it will be capital R, and for recessive, it will be small r, small r. And for the seed color, we can use capital Y, that is for yellow, and small y for green. Okay, let me write it down. This is brown, green. This is yellow, green. I hope you are clear with this. Dominant will be capital R, that is round, and recessive will be small r, that is wrinkle. And the seed color, we can use capital Y for yellow, and small y for green. Okay, now see, we are going to apply the same procedure that we did in the monohybrid cross. That is, we will find out the parent. Okay, parent, we will make a cross between round and yellow, wrinkle and green. Okay, these are the parent. Wrinkle, green. Okay, so for this round, capital R will be the genotype. And then capital Y. Okay. And the next step is we will find out the gametes. Since this is a homozygous case, okay, in dihybrid cross, in the homozygous case, we can take just one each of the character, one allele of the character. That is, from the round we will choose one allele. That is capital R, and for the yellow we will choose one Y. Okay, and here same goes. Okay, this is our gamete. So now we will find out the F1 generation. Okay, to get this F1 generation, we just simply make a cross of these two, just a simple cross. Okay, that is capital R, capital Y, small y. Okay, again, we can do it some more with this R and Y, you will get the same result. So, in order to avoid all the in order to avoid it, let's make it simple and then take this capital R, capital R, small r, and capital Y, small y. Okay, this is the F1 generation. Now after the F1 generation, this is all the outcome is brown and yellow. Brown and yellow is the outcome of the F1 generation in the dihybrid cross. Now the second that we are going to do is selfing. Selfing means we will make a cross of this to the same of this. Okay. Now let us see the second selfing. Okay. This is the outcome of the same selfing. So you will get the same capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Okay, this is the gametes. So you will get the same gamete over here when you make a cross of this. Okay. Now to do this, we require a Punnett score. For more convenient, we better do it in a Punnett score, which will give us the ratio of phenotype as well as the genotype.